Hello everybody, my name is Sam Williams. I'm at the University of Bristol and I'm now in my final year of my PhD. So in this quick talk, I'm going to talk a little bit about what we do at Bristol and a bit about an approach to bioprospecting that I've been working on for the past year or so. So the resource we have at Bristol, what we bioprospect, is a collection of deep sea sponges, around about 200 of them, from depths of up to 4 kilometers deep from the Atlantic. What I began to notice when we first started the project a couple of years ago was the massive variability you'd see in between these sponges. Some of them you'd get, 1,000 or so colonies, and the others maybe just a handful. And also the diversity would change a lot between each of them. Some of them would be completely dominated by a certain group of bacteria, and others would be, incredibly diverse. So I started doing a little bit of research into the sponge microbiome, and ended up reading a lot of microbial ecology papers. And it's a fairly hot topic of research because the sponge microbiome is in fact the oldest microbiome on the planet, and it's also one of the most diverse. So while I was reading these microbial ecology papers, I started to wonder if some of the techniques that they were using could be applied to the field of bioprospecting as kind of like a pre-screen before you start to culture them. And it would also help overcome one of our biggest problems, which was we had over 200 sponge samples, but only 2 PhD students, and we'd got through maybe about 6 or 7 in the past 2 years. So to cut a long story short, that's exactly what I did. Around about 25 different samples, 3 of them can be seen here. And it really did give us a fantastic insight into what samples we should be prioritizing based on abundance and other factors to take forward for screening. It also raised some interesting scientific questions, like, for example, what does a microbiome that is rich in antibiotics or rich in natural products actually look like? Does it look like microbiome A, which is fairly equally diverse and has a high number of actinobacteria in it? Or, tantalizingly, is it microbiome B, where a third of it can't even be assigned to a scientifically known phylum? Or possibly it's, microbiome C. It's completely overrun with proteobacteria, but is that because there's a super producer in there somewhere that's allowing the proteobacteria to proliferate and everything else to get knocked down a peg? I have cultured all 3 of these sponges, so hopefully I can go some way to answering that question by the end of the year. And, hopefully find some interesting compounds and antibiotics in the process. But it has really helped us screen our environmental isolates before they hit the agar plates. And I think it's a great way of prioritizing the work that you need to do. Thank you for listening, and if anyone's got any questions, please do get in touch.